Okay.
Yeah, thank you.
Good afternoon, everyone. It's the honor that, because of the COVID-19, we have to see each other online. Physically, we cannot see each other, but virtually, we can see each other and chatting with each other. And for those of you, just get online. And so the meeting will started, and I will introduce the first speaker, Dr. Kovit, because every time I don't know how to pronounce his last name, so I will skip that. I always call him Dr. Kovit.
It's okay lah.
Dr. Kovit have been in Siriraj Hospital in the Faculty of the Medicine in the Mahidol University for a long, long time. Just like I say, almost more than 40 years, I think.
Yeah, that's right.
More than 40 years. And so he had outstanding publication, more than 200 publication. And he has been serving as the chief for the Division of the Instrument for Research Office for Research and Development for a long, long time, from the 1993 until now, still to be the chief, okay? More than 30 years and counting, okay? That means that he had a very long career in the scientific community. This is means that he also get lots of the honor. It's a long list. You can think of everything, all kinds of honor. He probably get it. Okay, so without further ado, and I will introduce his presentation today. He's talking about the monocyte subset and the expression of the co-stimulatory molecule in patients with acute dengue virus infection. And that is what his presentation today. So Dr. Kovit is yours. Yeah.
Thank you, Professor Oscar. Hello, everyone. I'm very honored to be invited to give a talk online on the webinar during this very special occasion that we have the COVID pandemic. So we see each other online. So the topic that I'm going to present and share with you some of our findings. The title is under the title of phenotypic characterization of monocyte subset and the expression of co-stimulatory molecule in patients with acute dengue virus infection. Here is the outlines of my talk. I will divide my talk into 3 parts. The first part, I will give the background of the dengue viral infection, the distribution, classification, and immunopathogenesis. And then I will talk a bit about our finding on the dendritic cell in the dengue infection, as well as the monocyte paper that have been linked to the plasma-blast B cell differentiation. And finally, my talk will be on the title that I just give it to you. Which is a phenotypic chain of proliferate monocyte subset and their co-stimulatory molecule on immune response during acute dengue infections. Sorry. Oh, not go on. Sorry, the slide couldn't move. Oh, see. Sorry, this slide won't move. So I'm sorry, the slide won't move. Okay. So, okay, slide move now. Okay, so the world distribution of dengue in 2016, and you can see that the dengue distribution all over the world, but mostly in the tropical country, more than 100 tropical countries have been infected by the dengue virus. And out of these more than 100 countries, nearly 2.5 billion people are at risk of dengue infection. And of these is around 100 million people are dengue cases, and some of them have been hospitalized, and with quite a high number of mortality rate in the tropical area infected with dengue. In Southeast Asia, as you can see, all over the dengue distribution is all over the country, stretching from India to Taiwan. And you can see Taiwan also have the dengue. And in the southern part of China, Thailand, Vietnam, Cambodia, Malaysia, Indonesia, and Philippines, they all have the dengue cases. And some countries have more, some countries have less. And the dengue manifestations, we can divide dengue infection into asymptomatic and asymptomatic. Asymptomatic can be divided to the normal non-dengue fever illness and the dengue fever, which are very close, very similar to the flu. Dengue fever, it is without hemorrhagic. But the symptomatics of dengue virus infection ranging from dengue hemorrhagic fever to a very severe case that we call dengue hemorrhagic fever with shock syndrome, or DSS. And there are many immunopathogenesis theories of the dengue infections. The first one and the most people have been cited is an antibody-dependent enhancement replication, in which a person infected by the dengue serotype, let's say serotype 1, and their body memory B cell produce the antibody against the serotype 1, so-called neutralizing antibody. But this, and the person will recover from the dengue. And the memory B cell will gone, but leaving some very small number of the B cell, memory B cell. But if this person had been one infected by the same serotype, let's say serotype 1, the B cell, memory B cell will expand and proliferate into the antibody secreting B cell and produce the antibody and destroy the serotype 1. But if this person unluckily infected by the other heterotopic type or serotype, let's say serotype 2, the subneutralizing antibody from the serotype 1 cannot work. So this becomes severe. So and then with the binding with the FC receptor, they can go into infect the other cell, let's say monocyte, or other what we call Oscar have been found that megakaryocyte may be one of the dengue target. So once they infect this megakaryocyte or monocyte target cell, the serotype 2 virus expand and become very severe. And the other theory is what we call mast cell mediated vascular pathology. One the replication of virus become big, and the body build up the cytokine from many cells, for example T cell, and this cytokine, a lot of cytokine produced in the host to again the replicated virus we call cytokine storm. And this cytokine storm including TNF, IL-1, TNF alpha, and this lead to the vascular leak in the blood vessel. And so this we call mast cell mediated vascular pathology. The third one they call original antigenic sin. The model of original antigenic sin is very similar to antibody-dependent enhancement replication, in which a person infected by the first serotype, let's say serotype 1, B cell produce antibody, neutralizing antibody, and subside the dengue. But if this person recover and infected by the serotype of dengue, let's say serotype 2, the antibody B cell, memory B cell that produce antibody against serotype 1 cannot work because they are low number. But it expands because it cannot expand. So the B cell that should have produced the antibody against should work, but because they are outnumbered by the serotype 1 memory B cell, so that's why the virus can expand and proliferate. So this become very serious. Both original antigenic sins and antibody dependent enhancement produce a lot of virus, and this virus induce a lot of cytokines, so-called cytokine storm, that lead to the manifestation of the dengue infection. And so I'm going to share with you some of our previous work on dendritic cell and monocyte. Here is you know very well that in human immunity we have innate immunity and adaptive immunity, but today we will concentrate on dendritic cell and monocytes. And in the dendritic cell, we know that there are two types of dendritic cell. One is plasmacytoid dendritic cell, and the other is myeloid dendritic cell. Myeloid dendritic cell is the major population of dendritic cell, in which they capture antigen and present antigen to the T cell. Whereas the plasmacytoid dendritic cell that produce cytokine, for example, interferon alpha. And we know that dendritic cell normally during the latent state, we call them the dendritic cell is the immature dendritic cell. So they are co-stimulatory present expressed on the surface of the dendritic cell, but the number is very low. But if the person infected by the dengue, this is the danger signal. The danger signal activates the dendritic cell, and the dendritic cell become mature and express a lot of co-stimulatory cytokine. For example, CD80, CD86, CD40, and CD83. Monocyte is also antigen presenting cell, right? So when they see this danger signal, a lot of cytokine, for example, GM-CSF, IL-4, activate the monocyte, and the monocyte become immature dendritic cell with a low expression of co-stimulatory molecule. And this immature dendritic cell, they can go to and become mature dendritic cell, so-called immunologic dendritic cell, and the other we call tolerogenic mature dendritic cell. It depends on the expression of the co-stimulatory molecule on the surface. So this co-stimulatory molecule, for example, CD80, 86, they bind to cytotoxic T lymphocyte antigen protein 4. So they inhibit T cell. For CD80, 86 bind to co-stimulatory CD28 ligand. This activates T cell. The ICOS is inducible cytotoxic co-stimulatory ligand. This bind to the ICOS, and it can activate T cell or inhibit T cell, depend on the CD28 signal, and so on. So on monocyte, which is monocyte can differentiate into dendritic cell. They also express a lot of co-stimulatory molecule on the surface. But If it is a letting or naive monocyte, the expression of the co-stimulatory molecule is very low, unless it has seen the danger signal, for example, dengue infection. So, and because the advance of flow cytometry, we have 14 color flow cytometry. We can stain the cell, the white blood cell, the monocyte with antibody. And this antibody have been conjugated with color or fluorescein. Sorry, yeah. So with the flow cytometry, the polychromatic flow cytometry, we were able to stain the cell and look at the dendritic cell phenotype. And we can divide the dendritic cell into myeloid dendritic cell, which is the majority of the dendritic cell, as plasmacytoid dendritic cell. And we also have the double negative dendritic cell we call DN. So when we study the dengue infection by looking at the phenotype of the dendritic cell, we see that on the left-hand side, the majority of the dendritic cell is a myeloid dendritic cell, whereas on the right-hand side, you will see that in the dengue infected patient, the percentage of the dendritic cell population subset is different from the healthy people. And when we compare if dendritic cell subset, myeloid, plasmacytoid, and double negative dendritic cell, you will see that they are different. For example, the myeloid dendritic cell is low in the dengue infected patient when compared to healthy subject. And when we compare the dengue fever with dengue hemorrhagic fever, including dengue shock syndrome patient, you will see that the population, the percentage of these dendritic cell subpopulation, myeloid, plasmacytoid, and double negative. The myeloid dendritic cell is different between dengue fever and dengue hemorrhagic fever. And the other way around, the double negative of dengue hemorrhagic fever is higher than the dengue fever. And when we look at some co-stimulatory molecules, you will see that some of the co-stimulatory molecule expand on the myeloid dendritic cell is different from healthy and dengue fever and dengue hemorrhagic fever. And but some of the co-stimulatory molecule is not different between healthy and dengue fever. So this is very interesting because we can use the co-stimulatory molecule on the dengue or on the dendritic cell expression to differentiate out between dengue hemorrhagic fever and dengue fever. But we still need a lot of work to do because some we cannot differentiate, but some patients we can differentiate. Here come the other story, which is a monocyte. We can, like dendritic cell, we can divide monocyte into three populations. We know that the classical monocyte express CD14 and very low CD16. And a non-classical monocyte, which is the lower left quadrant, express low CD14, but a lot of CD16. And the middle one, which is an orange color, you will see that we call intermediate monocyte. And the majority of the monocyte in a healthy person is the classical monocyte, which is this gray color here. And accumulating nearly 90% of the monocyte population. The other two subsets are intermediate, which is orange color, and the green color, or which is the non-classical monocyte. They both combine together about 10% of the monocyte subpopulation. Here we have worked out on our normal subject and the dengue patient. We found out that very interesting that in the dengue patient, the intermediate, normally we have around 5% in the normal population, but in the dengue infected patient, the intermediate monocyte can go up to very high. Let's say some patient can go up to 12 to 15%. But as a whole, there are higher intermediate monocyte compared to the healthy subject. So when we study the transcriptomic or gene microarray, we found that when you look at on the figure D, the blue color and the yellow color on top represent the normal healthy and the convalescent patient from the dengue infection. And the red and the black color on the right-hand side, on the top on the right-hand side, here are the dengue infected patient. The red color represent the high viral load dengue patient. The black color represent the low dengue viral load patient. As you can see, when we compare the normal healthy convalescent person or with the dengue fever and dengue hemorrhagic fever, you will see the transcriptomic profile is different between a dengue infected patient and the healthy person. And so when we divide the dengue patients into high viral load and non-high viral load, you will see that they go very well with the non-structural protein 1 antigen of the dengue. They go together. And the finding also show that in the dengue, acute dengue, either is it dengue fever or dengue hemorrhagic fever, you will see that the double positive dengue, the intermediate, the dengue monocyte is higher. As you can see in the red color of the pie chart, on the acute dengue fever and dengue hemorrhagic fever, there are more intermediate monocyte. And you can see in the figure D that both low viral load and high viral load dengue infected patient had a high number of the intermediate monocyte at the middle figure, right? When compared to healthy donor. Yeah, and then when we incubate this monocyte, infected monocyte with the B cell, the B cell will be proliferate, as you can see from figure E. Then when you're using CFSE to label the B cell, and the B cell will proliferate. The B cell will be proliferate. As you can see that on the figure B, yeah, the red color and the black color represent the monocyte with the dengue, and when you incubate with the B cell proliferate more when compared to the B cell alone. And again, this monocyte dengue infected cell incubate with B cell, the B cell become plasma blast and produce antibody and produce cytokine, right? For example, many cytokine, which I'm going to show you in the next slide. So we found that in the monocyte dengue incubate with the B cell, they produce a lot of IgG, IgM, and IgA. So a lot of the cytokine, and this when we correlate this cytokine, for example, BIP1 alpha and P10, they correlate very well with the number of the intermediate monocytes. Okay, so the last topic I'm going to talk in my last 10-minute talk is about the topic of monocyte that we would like to share with you some of our recent findings. Here is the gating strategy. We use many color and many antibody to stain the monocyte, and then on the lower left figure, you can see that we can divide the monocyte into T subpopulation: intermediate monocyte, non-classical monocyte, and classical monocyte. This is the majority of the monocyte in the healthy subject. And then, as you will see, that we repeat the experiment, and we found that the intermediate monocyte, this is a figure C. You will see that dengue fever and dengue hemorrhagic fever have the high percentage of the intermediate monocyte when compared to healthy. And we were able to study the kinetic or longitudinal study of this population of the monocyte in the dengue patient. As you can see, for example, intermediate monocyte in figure F, they are very high during the febrile phase, okay? But they gradually decrease in the febrile phase. This is day +2 or day +1. The number of the intermediate monocyte decrease to normal. So we are able to study the kinetic of the subpopulation of the monocyte. And for co-stimulatory, we use antibody to stand for CD40, CD86, and ICOS-L on the monocyte and see what happened on the monocyte subpopulation. And we found that some of the co-stimulatory molecules are increased in the dengue fever and dengue hemorrhagic fever. For example, CD40 and CD86 and ICOS-L. These are increased in the classical monocyte and also intermediate monocyte. And when we look at the kinetic from the febrile phase to febrile phase, you will see that some of the co-stimulatory molecules expressed on the surface of the monocyte are decreasing. For example, CD86 at the beginning of the febrile phase, they are very high, right? But when the patient become convalescent and febrile phase, CD86 is very low, right? The same with the ICOS-L. So this is the kinetic change of the molecule of co-stimulatory molecule expressed on each of monocyte subsets. So in conclusion of our monocyte study, I can conclude from here that there are different patterns of T monocyte subset during acute dengue virus infection. Although there is no change in non-classical, we see the majority, we see the low number of the monocyte subset in the monocyte. And there are change in the other two monocyte. There are change, a significant change, decrease in classical. A decrease in classical monocyte, we see the majority of the monocyte, and an increase of the intermediate monocyte. As I said, some patients can go up to 15% from the original 5%. And we cannot use the monocyte to differentiate the different severity of the patient. So unfortunately, we cannot have that. So we need to have the transcriptomic to see so that we can differentiate from the severe case from the non-severe cases. And the different pattern of these two major monocyte subsets occur during early phase of infection, but they are gradually become normal. And for example, CD40, CD86, and ICOS-L during the forwardless phase. And we think that an increase in the co-stimulatory molecule on the majority of proliferable monocyte may indicate that this monocyte of dengue infected patient may have increased antigen presenting because they are expressed on the majority, the classical monocyte and intermediate monocyte. And the majority of the monocyte, they are the antigen presenting cell. So we think that they have increased antigen presenting function. That's why they show the high expression of co-stimulatory molecule signal and then the death signal of the dengue. So taken together, this data and may provide a useful understanding of the monocyte subset distribution and their kinetic based on their frequency and co-stimulatory marker during the course of dengue infection. So with that, I would like to thank my colleague, many of my colleagues, and also thank you, Professor Oscar Perng from the National Cheng University, who have been working with us for quite a number of years. And I would also like to thank Professor Kunkanya Chokpai from the Department of Pediatrics, who is in charge of the dengue patients for us. And also would like to thank Professor Ansari and Professor Ahmed of the Emory University. And also thank for the NIS grant to allow us to carry on this project, and also the Thailand Research Fund. Thank you. Thank you.
Okay, thank you for Dr. Kovit's presentation. I just see quite a few of the questions that have been posted up in the group, and some of the questions that I think we probably will wait until my presentation and we can answer them one by one. Okay.
Okay.
So I will go ahead and start my presentation first, and then we come back to the Q&A. Is that okay?
Okay.
So can you see my slide?
Yes.
Yes.
This is my title of the presentation. I introduce myself, okay? And people call me Oscar. I have been used to be at Emory University, where I also working on dengue, and come back to NCKU about 7-8 years ago, and continue working on dengue. And so that's about it. And so without further ado, the reason I put on this title, because I try to get an overview of the dengue virus infection, and there are many questions that hasn't been answered, and I try to answer some of them. Okay. So as Dr. Kovit already mentioned, and so there are dengue mainly distributed in tropical and subtropical area, but because of the climate change, it has been spread all over the world, and so the burden of the disease has increased for many times. It is estimated that every year we do have almost 400 million people got infected, and which is resulting in about 20,000 related deaths every year. And the increase of the traveling, that is before the COVID-19, and so increase the spread of the dengue virus. But we are not sure after the past 2 years of the COVID-19 lockdown, the dengue case will go down significantly globally or not. I'm not sure about that, but I'm pretty sure in the dengue endemic country, the dengue is still there, and probably as high as the COVID-19. Probably, we don't know. But the dengue does not cause the death as much as the COVID-19. Okay. So the transmission cycle, everybody knows that is. Originally, dengue is coming from the jungle, from the monkey, and we visited the jungle and got bitten by the mosquito that happened to carry the dengue virus, and human carry the virus into the city and become the urban cycle. And the main mosquito is the Aedes aegypti, transmitted the dengue virus. And of course, recent evidence to show that blood transfusion, organ transplantation, bone marrow and stem cell transplantation, mother to infant, also can transmit the dengue virus. Okay. And so we spend a lot of the time in dengue fever and dengue hemorrhagic fever and dengue shock. So most of the time it's only about very low percentage of the people. It's about 1/4 of the infected people will develop either dengue fever or dengue hemorrhagic fever associated with the dengue shot. But 3/4 of the infected people are mainly asymptomatic, okay? So we spend lots of the time and money to research why certain people develop DF, but other people develop dengue hemorrhagic fever. As of today, we still really don't know what are the exact mechanisms are, okay? But exact mechanism, why certain people develop dengue hemorrhagic fever, okay? And we also don't know why only small percentage of people will present the clinical symptom, okay? And as of today, yes, we do have the approved dengue vaccine, but the efficacy is concerned. And as of today, we haven't really have the antiviral drug yet, okay? And recently, they do have another avenue that associated with dengue, we call the congenital dengue. It has been shown long time ago, but we really, there's a hard study on it. And I'm going to present a few of the studies that we already done. And as I mentioned, majority of the people are asymptomatic. But this group of the people, we hardly spend effort to study them. We don't know why, but maybe because of they appear to be normal, healthy. That's why we don't study on them. But evidence shows that if these people donate the blood or organ to the healthy people, to the recipient may develop the dengue fever or dengue hemorrhagic fever. Okay, so this maybe suggests that this kind of asymptomatic people may become the persistent infection or become the carrier, and maybe play a role in the dissemination upon bitten by the mosquito. But this is the research that we hardly really work on it. Okay, so this is one of the avenues for those of the young people who would like to study dengue. You probably can look into that. So as I mentioned, and also Dr. Kovit mentioned, that we spend lots of the effort to try to understand, to differentiate why certain people develop dengue fever, certain people develop dengue hemorrhagic fever, or dengue fever will slowly progress to the dengue hemorrhagic fever or not. There are two different kinds of hypotheses. And so some people say that yes, some people say that no, dengue fever and dengue hemorrhagic fever are, each one of them is unique disease. Okay, it depends on which point of the view you're looking at. Okay, because so far we don't have the very good animal model to study this kind of disease. So that's why everybody say that he say, I say. Everybody presented based upon the evidence that they already collected. So as Dr. Kovit also said that the cytokine storm is one of the potential mechanisms that contribute to the dengue hemorrhagic fever, which is manifested by the capillary leakage. Okay, the blood vessel become weak, so become leakage. Okay, so we in Taiwan we don't consider ourselves as a dengue endemic country because most of the people that we have the dengue, most of the dengue patients are primary infection. So we presented this kind of results in recent publication. We did we call the cytokine panel. So each sample we blast through 23 panel of the cytokine, which means that lots of the money invested and come up this paper. So at the end, what we found is that IL-10 would be a potential diagnostic marker for EF. And CD121B would be the predictive of the severe dengue. So, okay, this is not in the endemic country. This is the result from the dengue non-endemic countries such as Taiwan, okay, which is not as severe dengue as the Vietnam, as the Thailand. We don't know this can be sustainable in a dengue endemic country or not. Okay, if you're interested in, you can go to read this article, which is just been published this year in March. And in terms of the permissive cell for dengue virus infection, as Dr. Kovit already mentioned, that probably some people seen that monocyte or macrophage could be infected, but in in reality, they can be infected, but they may not produce infectious dengue virus. So over the year, that we did is that we found that the megakaryocyte lineage all the way to the stem cell, all the way to the multiproton progenitor cell. This kind of lineage is preferred target for the dengue virus. Maybe that is one of the criteria for the thrombocytopenia occur in dengue patient, in particular in severe dengue. So these are the papers that over the year that we published, including this year April, we published paper in Harrier, which talk about the stem cell in the cord blood. Okay, I'm going to present some of them. So all the way, this is the lineage, the lineage that we think that the preferred target by dengue virus. And again, I say this is happened in the primary country, primary infection, non-endemic countries sample that we over the years that we work it out. But we are not sure in dengue endemic country where experience each person being exposed to the dengue virus infection multiple times, where this case is sustained or not, we don't know. Maybe we'll wait for some other people to confirm for this kind of finding. So this is been published in 2 years ago. So we found that the stem cell are easily to be infected. The markers such as CD34 or CD133 in the bone marrow, that if you isolate them, you do the infection, you can see them. These cells are very easy to be infected. And we also found that these stem cells being infected, they will not die, they will survive. And so they do have to go through the, we call the stress period, such as 12 hours and 24 hours. And after 36 hours, they go back to normal. And they do have this kind of spike formation. And this, if we look at closely, they may check in some kind of vesicle, okay? But they are perfectly normal and they do regain the function of the stem cells, such as if you perform the sulfidical assay or colony forming unit, and they're still functional. Okay, so, and then if you take out this cell and you do the culture, you can recover the dengue virus. Okay, so this is what we've seen, that the stem cell become one of the preferred target for dengue virus. The reason why, because the stem cell is immature in their innate or immune system. So that's why the dengue virus take advantage of this kind of cell. Maybe that is one of the reasons why maybe stem cell become vulnerable to be infected by dengue virus. So this is the reason why we think that dengue might be able to get along with the CD133 positive cell after they are being infected, after this cell being infected. Okay. So this means that the dengue virus stay inside the stem cell. What can they do? So this is the next slide, which have been published in this year, that we can recover the dengue virus in this, we call the infected stem cell. So if you do the co-culture, take out this cell, you do the co-culture, which means that you take the cell in the indicator cell, such as viral cell, and over the day, you collect the supernatant, and you can look at the Panelist, and you do the viral titration, you can see the virus being recovered. So that means that even though in the supernatant infected original CD133 infected supernatant, you don't see the dengue virus, but if you take out this cell, and you can recover the dengue virus. So that means that the CD133 or 34 would be the reservoir for dengue virus. That is the finding that recently finding. So because of this kind of things, we found, and then we think that stem cell could be the reservoir for dengue virus, maybe for a long time. We don't know how long, but quite in recent years, there's many evidence to show that if you do the stem cell transplantation in the people who are visiting the dengue endemic country and do the stem cell transplantation and then recipients are easily to be infected by dengue virus. So this is one of the reasons why maybe stem cell transplantation can transmit the dengue virus in the healthy people, in the recipients. So this is all other things collected. So with our finding, we see that dengue virus stem cell could be the target cell and also reservoir for dengue virus. And maybe this is one of the reasons why, we stem cell being targeted by dengue virus. And so this could explain why maybe a typical lymphocyte in dengue, because how does this typical lymphocyte really form? There are many different kinds of hypotheses. And some people think that it's a cytokine induced them, but we think that probably it's the origin of the stem cell being controlled, handcuffed by dengue virus, and resulting in the differentiation difficulty. So maybe produces the typical lymphocyte. So this is where is the evidence? So this is what we think that maybe that is the reason. So for the stem cell, and if they're going to become the downstream for differentiated or proliferation, they require the two major transcription factors called the GATA1 and GATA2. These two transcription factors require to be balanced. Either one of them tune into GATA1, you will get one lineage. Tune into GATA2, you will get another lineage of the cell. So such as GATA1 expression expressed in the stem cell, you will favor to the megakaryocyte or erythroid cell lineage. But if the GATA2 being downregulated, so it will become the initiator cell differentiation. Okay, so this is the transcription factor. You can see that after dengue viral infection, stem cell, SC is a stem cell, is really up and down, but the most affected on day 5 and day 7, and early megakaryocyte progenitor cell being infected in later stage. And amazing, we also find the GATA3 also increase. Okay, this GATA3 mainly is preferred to the lymphoid lineage. So this is the things that was upregulated during the dengue virus infection. So, and GATA3 is the master regulator of differentiated T cell development. So if it, it sets GATA3 above the T cell development. So this is the reason why we see that the stem cell being infected, and then if you look at the downstream colony myeloid progenitor cell or inhibited, okay, that means that all the downstream are being dysfunctional, not really as effective as the normal cell. Okay, they do have some certain preferred lineage that stem cell go into. So overall, you look at this slide, if we found that the GATA1 increase, GATA2 also increases, the balance outcome become to inhibition of the common myeloid progenitor cell, resulting in the myeloid blast decrease, and later on become the monocyte decrease. But at the same time, you may increase the megakaryocyte lineage. Okay, but the GATA3 increase prefer lymphoid lineage. Maybe this is the outburst resulting in the plasma blast that we see a lot, or a typical lymphocyte. We don't know, but this is one of the mechanisms that need to be worked out in the later on, in the future, for those of people, the young junior scientists who are interested in this kind of dengue research. You can find out how does a typical lymphocyte really, produce in dengue patient. So our finding, they do have the potential outcome. Maybe that is can explain why dengue fever can mimic in plasma cell leukemia. If that is potential, this is the case report. And recently, in our population-based cohort study, we found that people who are being infected by dengue virus, and they are likely to develop leukemia after 3.5 years. Okay, this is one of the, we call the cohort study in our National Health Insurance Register program, because we can know who get what, and so we take out the data and then do the heavy analysis, and we are able to find that the people who are being infected dengue virus are likely likely to have the risk of leukemia after 3.5 years. Okay, maybe that is the reason why the stem cell doesn't have the virus when we need the stem cell, and dengue virus control the stem cell differentiation and proliferation. That is one hypothesis, but actual mechanism we haven't worked out yet. Hopefully one day we will work it out. Okay, and come back to the asymptomatic dengue virus infection. As I mentioned, they are accounting for 75% of the dengue infected population, but very, very few evidence to investigate what is the potential role of the symptomatic dengue virus infection subject. Okay? So asymptomatic or persistent dengue virus infection have been proposed a long time ago. In 1939, people already found that, you know, some of the healthy people, if you transfer their serum to another people, the recipient can develop dengue virus. So at that time, they call the dengue in appearance people. That is a long time ago, but people did not follow up. And also potential viral latency being proposed by Dr. Hada. Dr. Hada is the person who is the first person to isolate the dengue virus. Okay, so Dr. Hada seen that potentially there is a viral latency in the human body. Okay, but we haven't really studied a lot on it. So we would like to take a look at some of these. And so very recently, people proposed that asymptomatic human would transmit the dengue virus to mosquito. Okay, so this is one of the challenges here. Asymptomatic can be the incubation period, or can be the people who just become asymptomatic regardless of what happened. Just like the COVID-19 case, quite a few people, they appear to be healthy. If you do not do the screening, you don't know whether they carry the virus or not. The same, we see that the same kind of scenarios also happen in dengue virus. So some people may carry the virus, but they appear to be healthy. But when mosquito biting these people and transmit to other people, and other people become clinical symptom. Okay, so there may be, that is a potential, but this require massive amount of the public work, and we look into this some of the things. And so recently, it is estimated that probably dengue outbreaks are mainly coming from these asymptomatic people. Okay, because they are they call the silent majority, you know, transmission. So maybe these people accounting for majority of the dengue outbreak. So this is one of the area that hasn't really been really in field confirmed yet. Okay. So we also take advantage of the 2015 when we had the big dengue outbreak. We try to estimate how many people who are asymptomatic dengue viremia after the, you know, a big outbreak. So we look at quite a few people at the end stage of this pandemic, and we found that we estimate that on average it's about 10,000 people. There are 15 people are potentially carry the virus. So if these people do the blood donation, then maybe they do contribute a lot. Okay, because each blood component you can make into about 1.45 transfusible components, so it amplifies a lot. Okay, so this is our estimation, and so we go on to do the field site collection. So we collect the 1,250 people, healthy people, and we do the ELISA assay. We found that we do have the IgM positive, almost 5% or 8% respectively, and that means that we are not the dengue endemic country, we are, mainly primary. But we found that they do have the NS1 negative. And in all, we found that we do have one person, we do have the RT-PCR positive, one out of this 1,200 people. But if you do the SD dengue rapid test kit, NS1 are negative. And because of this, we do isolate this virus. We send it to a company to develop the new dengue rapid test kit. We call the asymptomatic dengue rapid test kit. And after that, you use the same serum, you can see that the dengue NS1 can be detected. Okay. And these people, this individual do have the very high viral load, can go up to, in the serum, both of the serum and plasma can go up to 10 to 5. And because of this high viral load, we can also subject it to look for viral particle in this serum, because like I say, in the field of the dengue, even though we know that dengue has a viremia, but basically there is no report what kind of the viral morphology in dengue patients' viremia. So when I first asked this question, quite a few people asked me, is that important? Of course, it is important, because nobody look at the, we call the actual viral morphology. So we take advantage of the 2015 of the dengue outbreak in Taiwan. So we found that dengue virus viral morphology in acute patient is very, very unique. It's just like a, sunny side up egg morphology, just like the, morning egg. It's very, very unique. People who are interested in unique viral morphology, you can refer to the paper that we published in 2015. So this high viremia asymptomatic patients' viral particle, we also subjected to EM image, and we found that it's similar to acute dengue viral morphology that we published in 2015. So the dengue viremia, the viral particle is very unique in dengue patient. That means that the epitope presentation may be different. And if we use, the tissue culture-based viral particle, maybe the neutralizing titer will be different. So that is the reason why in dengue field, people always say that you have the neutralizing titer. Doesn't mean that you are protected. So because of the viral particle in patient is different from the tissue culture viral particle. Okay, that is what one of the hypotheses that what we see. And we would like to know how many people really could be, the potential healthy carrier from the dengue endemic country. So we screening quite a few people, and we are able to capture 8 people with the, we call the asymptomatic dengue rapid test kit. We are able to find 8 people are NS1 positive. Very interestingly, these people, although these people are from dengue endemic country, quite often we don't see the dengue IgG or IgM. Only in one people we see the dengue IgG, and one people we see the dengue IgM. But these 8 people are all NS1 positive. So if we look at where does this NS1 located, we found that mainly in the stem cell. So this in such as the CD133 or CD34. Okay, so this asymptomatic people's NS1 positive, mainly we found that likely associated with the stem cell in the peripheral blood. Okay, and to confirm this, we established the humanized mouse model, and we do found that the dengue can become latent, and the virus can be reactivated again. And so if you look, the mice can be survive for almost a year. We monitor them to the 223 days, which in this period, they do have the 3P, the virus come back, okay? And at the end, we sacrifice, and we also take out the virus from the stem cell of the bone marrow. We do the co-culture, and we also found that the bone marrow stem cell, the CD34 positive cell, we can recover the dengue virus. Okay? So suggesting that the dengue virus really hiding in the stem cell. So we think that this asymptomatic carrier probably after each outbreak will increase the population. After certain cycle of the dengue outbreak, the asymptomatic may be widespread in the whole area, and the dengue virus probably difficult to remove, and that means the reason why we can see that dengue always be year-round in dengue endemic country. That is one of the hypotheses that we throw out there. Hopefully that people can testify in the future. So at the end of the summary, that my presentation will be that we think that dengue virus infect the specific cell, such as the CD133 positive cell or CD34 positive cell, and they will become reservoir in this cell. And they do potentially do have a no productive phase, we call the latent infection, and they may potentially reactivate and transmit the virus to the healthy people. So this is the model that we are working on for us many years, and hopefully one day we can really figure out that this is really happening in human beings. Okay. So I would like to thank you for collaborating. Of course, thank you, Dr. Kovit as well, and also the people who are from the Kaohsiung Medical University and NCKU, those doctors who provide us the sample, and also my student, and all this work are mainly from the PhD students, such as the Brett Wang, Henry N, Chen Ho, Erwin Park, and Amrita Beth, and also the master student. And thank you for the Taiwan MOS grant and NHLI grant and CDC grant to support this research. So my presentation end up in here, and so now we can answer the question. And so how can I go back? Hello?
You may stop sharing, Professor.
How can I go back? Stop sharing. Okay, now.
Yeah.
So now
Okay.
I request that everybody turn on your camera so that we can take a group photo before we answer the question. Can all the participants turn on your camera so that we can see your face? Good. Hello? Can everyone stay online, still online? Can turn on your camera? Can everyone turn on the camera? Did you send the message to everyone so that we can turn on
Yes.
the camera?
Yes, please.
None of my students turn on their camera. Huh? Hello? let me enlighten them. Yeah. Please ask them to.
It's okay lah. Okay lah. It's okay. It's okay.
来猜一下，看你们在哪里。也有我的朋友，也有我的朋友，也有我的朋友。
Dr. Kovit. Dr. Kovit. Hello. Dr. Kovit already did the podium?
Maybe he have some problem in internet connection.
Ready?
Yeah. But I have his photo already, so maybe you can take it first.
We can go ahead and then take the photo anyway, because all my students.
Yeah.
There are quite a few people, but they do not turn on the camera.
Okay, maybe when I say 1, 2, 3, maybe we smile together and I take the picture. Thank you.
Okay.
Okay, 1, 2, 3. Okay, one more time, please. 1, 2, 3. Okay, I have done it. Thank you so much.
Thank you. Yes. So there's the second page, is that right?
Yeah.
They have the second page.
Yeah, already, Professor.
You already take it. Okay. So now we can answer some of the questions that people posted in here. The first question is what are the molecular biology testing technique used in this study and what are their function? So in Dr. Kovit's presentation, mainly is we call the flow cytometer, which means that you can use a specific antibody to a specific marker that you can differentiate them, which one is the T cell, which one is the B cell. This is we using the flow cytometer, okay, so that you can differentiate that all different kind of cell and identify their potential function. Okay, that is one of the question. The first question. The second question will be, two people are dengue infected. One has more severe symptoms than the other after both of cure to the one with a stronger immune system have a lower risk of the leukemia. We don't know. That is the good question, but we don't know because this require people to figure it out later on. Okay. The third question will be, which other dengue non-structural protein reduce antibody secreting cell in hematological disease, such as NS2, NS3, NS4? If so, then what is the ratio between its impact and that of the NS1? I am not sure about this because most of the people see that NS1 is one of the indicator for dengue virus diagnostic marker, and their function have been shown that engage in the viral replication. So does NS2, NS3, and NS4. But whether their function really related to the antibody secreting cell or not, we are not sure. Because in my knowledge, I do believe that there is no report on this kind of study here. Okay. And another question is, it is said that adults suffering from the severe dengue virus are more dangerous than the children. It is right or not? Yes or not? Because yes, there's some report talking about this, but still others think that they are both similar to the children, either your adult population or the children population, and maybe the severity are very similar. Some people think that there's no difference, but some people think that there is a difference. It depends on where does it come from. Like in Taiwan, yeah, just like I mentioned, most of the people are primary infection. So maybe the adult population is much more severe than the children. But in dengue endemic country, most of the affected population are children compared to the adults. So maybe that is the reason why children are much more severe once you get the dengue virus infection. So it depends on where does it come from. Okay? And the next question will be, do reinfection usually resulting in more severe clinical manifestation, either by the same serotype or different serotype? This is the same thing, you know, some research also show that the same serotype can induce severe dengue. Like if you refer to the EFAM's research in Kampapet in Thailand, where they to demonstrate that people being infected dengue-3 a year before, the second year still being infected by dengue-3, the same serotype, and the disease also kind of severe. Okay, and so most of the people believe that different serotype may introduce much more severe disease, but these are all based upon the dengue endemic country results and epidemiology study that, you know, in the country where most of the people being infected, and so it's kind of difficult to say whether they are more severe clinical manifestation or not. Just like I say, we don't know why some people will develop DHF, some people only develop DF. So this is so far, it's $1 million question. So we don't know why yet. Okay. And the next question will be, what are the symptoms that patients who recover from dengue infection are likely to have the leukemia after 3 to 6 years? We don't know because we haven't really go out to look for these people yet. Because the presentation that we have is based upon the cohort study, that is a retrospective data mining study. So we don't know that what kind of the symptom that people may have, but we think that may likely that the bone marrow disorder maybe is one of the criteria, but we don't know yet. Okay. And the next question will be, usually dengue patient will be checked for platelet count frequently. Does that directly correlate to the possible development into DHF-DS, or the platelet number is just one of the consequences of the dengue infection? So correlation-wise, people know that if you do have the low platelet count, yes, you will likely to have the dengue hemorrhagic fever and dengue cell. That is have been well established. But the mechanism leading to the low platelet count, there are different kind of scenarios. Some people think that is the autoimmune. Some people think that destroyed directly by the dengue virus infection. Okay, so and some people think that the thrombin goes down, induces the platelet count goes down. Okay, so it depends on which one, which aspect you're looking at. Okay, so but the correlation is there. If you do have the low platelet count, you're likely to have the dengue hemorrhagic fever and dengue shock syndrome that have been well established. Okay, but the mechanism, it have a different kind of hypothesis. Okay, and that's it. So all these questions that people posted, and I answered some of them I really don't know, but hopefully that after the presentation, you will learn more about the dengue virus, even though they don't kill people as much as the COVID-19. However, the economic impact is still there. Okay, if you're not careful, people still dying. Okay, so that is the bottom, the message that is the total message that I would like to deliver. Sure. How you have others to say? Dr. Kovit, do you have any others to say?
I didn't see any question for me.
You have to go to the chat question.
Chat.
You go to the chat, you will see there's a positive question on that.
Maybe Someone asked the question and then I can answer the question right away. I didn't see the question.
The question for you will be what are the molecular biology testing technique used in your study and what are their function?
Okay. Sorry, just no disconnected. Thank you. So, what are the questions, Hian? Dr. Hian, can you
Question
read
for
the
you
question?
is people asking is what are the molecular biology testing techniques used in your study and what are their
Okay.
functions?
So we use gene microarray from looking at the transcriptomic patterns of the gene presence on the cell. We use mononuclear cell, and then we extract the nucleic acid, and then we send out for the transcriptomics analysis at the Emory in the US. And yeah, as you can see from that paper, you will see that the dengue patients, either is it dengue fever or dengue hemorrhagic fever, the pattern of transcriptomic is different from the healthy or convalescent patients. So this might be, but we haven't looked into the detail of each of the transcriptome to see which protein or which RNA are involved and can differentiate between the severity of the patient. So we haven't do that. We haven't done that. Yeah. But what is a good question? And up to now, like Professor Oscar says, no one have no which marker or which biomarker is the most important. Somebody like Dr. Oscar says IL-10 maybe can pinpoint to the dengue fever, but not dengue hemorrhagic fever. Yeah, in our immunophenotyping, again, we can still not differentiate the dengue fever to dengue hemorrhagic fever. Yeah. Any more questions?
I think that's some of them are already answered, but I'm not sure in the audience whether they do have other question or not.
Yeah, in one of the questions in which Professor Oscar had already answered is whether the heterotopic serotype, the second infection, contribute to the severity of the dengue. Yeah, many people believe that the second different type of the dengue serotype confer to the severity of the dengue. But like Waka says, there is some papers that even if it's the same serotype, sometimes they can contribute to the severity of the dengue. It's very important that because Vietnam, Thailand, we all in the endemic area. When you take the blood, and you will see that we all have been infected by some certain serotype of the dengue, right? Maybe in my body, I already have 2 or 3 serotype already. But as you know, the asymptomatic is outnumber the symptomatic patient. So it pretty much depends on the host immunity of the host. Although virus itself can contribute to the severity, but a lot of the protection is come from the host immunity. Yeah. Yeah.
So Dr. Chuen,
Yes.
do you have anything to say? If not, we're going to conclude the meeting.
Yes, please. I don't have any question.
Dr. Hien, did you take the photo already?
Yeah, I have your
Yes.
photo already. Yeah.
Oh, okay.
Yeah,
Because
thank-
just now my notebook didn't work. The connection had been out, so I have to reconnect. Are
Yes.
you sure I have the picture inside?
Yeah, I have the
You need
picture.
you need to get a new computer just like me for the purpose of this
Oh,
meeting.
this is the notebook from my student. I forgot to bring to the office today, so.
Because of the COVID-19, I got a new computer because for this purpose.
Okay, good for you.
Okay, thank you very much for everybody to participate in this webinar, and hopefully that we'll see everybody soon.
Okay, take care.
Yes,
Thank
Dr.
you.
Meeting, is that right? Yeah, bye-bye. Thank you,
Bye.
Dr.
Dr. Hien, goodbye.
Goodbye.
Goodbye. Thank you
Professor
very much.
Oscar, take care.
Hey, see you, Dr. Kovit, see you online, okay?
Online,
Good
bye.
to see you. You never look old. You never look old some way.
Okay, still.
Okay, bye. Okay,
Bye.
see you soon. Bye. Okay.
Bye.
Dr. Him, bye.